fundamental question in biology is how a single genome can give rise to multiple cell types and lineages. More specifically, my lab is interested in immune cells, and our goal is to find how all cells have identical TCGA combinations, and the cells of the immune system utilize them differently and give rise to these distinct patterns of innate and adaptive immune response. The answer to this question, like many other questions in the field of epigenomics, there will be some redundancy between what Nick presented, but our work is mostly focusing on the histone modifications rather than the chromatin accessibility. So superimposed upon DNA sequence is an epigenetic layer containing key information specific to every cell type. Very briefly, DNA is wrapped around 8 nucleosomes or 8 histone proteins or nucleosomes. Epigenomics refer to the collective state of DNA methylation and also covalent modifications of histone proteins. These are basically the tails of histone proteins. These covalent modifications are closely related to accessibility of chromatin transcription factors to DNA, like what Nick was suggesting. Open chromatin sites are typically flanked with nucleosomes with unique combination of histone modifications. For example, open chromatin sites that are close to genes are usually flanked with nucleosomes where on histone lysine 4 on histone 3 can go under 3 methylations or H3K4 trimethylation. And measurement of this particular modification has been a great indicator of presence of open accessible chromatin site at that region. The key point here is that open chromatin sites are typically regulatory elements controlling gene expression. Over the last few years, we have learned that even different categories of regulatory elements can be differentiated and classified from one another based on their unique combination of histone modifications. For example, what I described to you in the beginning, promoters that are regulatory elements close to transcriptional starting sites are typically associated with H3K4 trimethylation. In contrast, distal regulatory elements that are away from genes, and as Nick very nicely presented, are called enhancers, are typically associated and can be very reliably delineated across the genome by binding of proteins that are able to acetylate the histone or histone acetyltransferases such as P300, or measurement of the histone acetylation level itself such as H3K27 acetylation has been shown to be a reliable way of finding the exact genomic location of enhancers. So many of these studies are now been functionally proven. For example, the binding of acetyltransferase P300 has been this is a study from 6 years ago, but they performed a very comprehensive study by generating a large series of transgenic mouse, and they showed how tissue-specific binding of P300, which is again a histone acetyltransferase, can reliably predict the functionality of those pieces of DNAs being functional in that relevant tissue. So with this wonderful discovery, finding the chromatin signature of active enhancers, variety of maps of enhancer elements have been generated in many cell types, tissues, in primary cells, cell lines, in both human and mouse. And what is really emerging, and I think was very nicely demonstrated in Nick's talk, is that, first of all, if we put together all of these maps, what emerges is that there seems to be about 1 million or so regulatory elements or enhancers embedded in the human genome. But what emerges by comparing all these maps is that each one, only a subset of these regulatory elements, tens or thousands of them, are active in a specific cell type in a given tissue under, especially within the context of immunology, under a very specific immune activation. So with this introduction, I just want to tell you what the work that I did as a postdoctoral fellow at John O'Shea Lab. So we had a very simple question. We wanted to know if we focus on CD4 plus T cells and the T helper subset that differentiate from the naive T cell. At the time, it was definitely not very clear the extent of difference in enhancer landscapes, but we wanted to know how similar or different the enhancer landscape were in T helper subsets. And our second question was, what factors are really able to make those changes happen in T helper subsets? So again, we focused on CD4 T cells, a little bit a kind of introduction on naive T cells differentiated to a variety of T helper subsets. We focused on T helper 1 and T helper 2, a few description of the key players that make this specialization happen. The changes in the cytokine environment, which is the reason that T cells differentiate and become specialized cells to take care of distinct kind of pathogens, start through the cytokine being expressed in this extracellular environment. These changes are transmitted to nucleus by proteins that are called STATs, and obviously there are multiple STATs responsible for differentiation of different T helper subsets. And then we have another class of transcription factors that have been historically called master regulators of each subset, but we have reasons to believe that maybe we should reconsider how they are master or not in this particular context. But these are transcription factors like T-bet in T helper 1 and GATA3 in T helper 2 that are endogenous, so they are expressed after they receive the cytokine, presence of the cytokine in the environment through STATs. And they are lineage specific, so T-bet is specific to Th1 and GATA3 is specific to Th2. So again, very reiterating the questions that we had, we wanted to characterize the enhancer landscapes in Th1 and Th2, that within the last few days they were the same cell type, they were naive T cells, but we wanted to know how different the enhancer elements are between these two cell types. And the second question we had was whether this first category of transcription factors are responsible to making the change in the enhancer elements, or is it about these endogenous transcription factors that cause these changes in the enhancer landscape? So we generated P300 map of P300 binding in Th1 and Th2 cells, and again, I'm pretty sure you're not surprised to, after Nick's talk to hear that about 50% of the enhancer elements mapped by P300 binding were distinct to these different lineages. And again, these are, going back to the tree of cells, these are really the bottom of the leaves, and they are typically CD4 positive T cells, and their only differences is they are able to express distinct kind of cytokines, yet they acquire about 10,000 unique regulatory elements once these cells started to differentiate. The overlap between T cell and macrophages was very small, and even very small number of regulatory elements were shared between mouse embryonic stem cells, T cells, and macrophages, and again, highlighting the fact that enhancers are incredibly cell type specific. So what I showed you about the P300 binding is, again, a representative of active enhancer elements. But we also look at another enhancer mark, which is HTK4 monomethylation, which is an indicator of poised enhancer elements. And again, it was interesting to see that the poised enhancer landscape between Th1 and Th2 cells were quite similar. What was different was only a subset of these regions further got activated with presence of the appropriate cytokine. So again, the poised landscape is similar, but the the gain in acetylation is what is unique, and that is eventually dictated to gene expression. When we generated these enhancer maps in T cells, there were some parts of the genome that were quite interesting and had really fascinating properties. I don't know if you can see, this is IL-2 receptor alpha, and there were these parts of the genome that had two very distinct properties compared to the rest of the genome. First of all, the y-axis, the intensity of P300 binding was quite strong and exceptionally different compared to the rest of the genome. And also these regions were kind of a cluster of regulatory elements. There were many regulatory elements, again, profiled by these P300 bindings side by side, at the vicinity of these important genes that we did know are key players in T-cell biology. This was very similar to the map of population of Manhattan, where, again, we have this huge concentration of everything being within this small piece of land. So thanks to Rick Young and Francis Collins, these regions were characterized as super or stretch enhancers. Again, it really doesn't matter what we call them. There seems to be some genomic regions in every cell type that harbor a large portion of the any signal associated to transcription apparatus, and they have fascinating properties, including that they happen at key identity genes. Again, they can be characterizing super enhancers can be a way to find the important genes in cell types that we have less or limited knowledge. They are sensitive to drugs, and it seems that the genes that have these kind of genes are more sensitive to any kind of perturbations. And a very interesting observation that we had, and it was also replicated in many different systems, was that the genetic variants associated to the super enhancers in cell types, in particular cell types, are typically enriched with genetic variants that cell type is known to cause those kind of diseases. So we're simply interested in finding what are these regions in the genome of T cells. So we characterize super enhancers in Th1, Th2, Th17, and we learned that even the pattern of super enhancer in T cell subset is even more cell type specific compared to the whole collection of enhancer elements in these three cell types. And not only that, other interesting patterns emerge. It seems that these regions, these non-coding part of the genome, are also highly transcribed. About 1/3 of the whole collection of non-coding RNAs that exist that are expressed at some point in CD4 T cell during CD4 T cell differentiation have a characteristic of a super enhancer, and by looking at the gene expression of T cells during differentiation, we learned that the non-coding RNAs that overlap with a super enhancer structure are highly dynamic. Their expression changes during T cell differentiation, and also the expression is incredibly lineage specific. And our other question was, so who are these super enhancer genes in T cell subsets? What was very interesting and emerged from the gene ontology analysis was that in T cells is all about cytokines and cytokine receptors, as one would have expected. In contrast, we have in macrophages, we have the chemokines, and in mouse embryonic stem cells, these super enhancer genes are mostly associated with transcription factors and factors that are important for repressing the opposite lineages. I told you that based on gene ontology, we realize cytokines and cytokine receptors are mostly harboring super enhancer features in the genome of T cells, particularly T helper subsets. But when we ask who is really the number one in terms of, again, harboring the largest amount of signal, who is the highest skyscraper of Manhattan in the genome of T-cell subsets. And this was surprisingly not a cytokine receptor. This was a transcription factor. And indeed, so this was BOC2, which is, okay, I can't go back. This was a transcription factor called BOC2, which is a repressor of which is a transcriptional repressor, and in a collaborative work with Nicholas Stifo, we showed how BOC2 is important in repressing the effector function and really fine-tuning the expression of a variety of effector genes and putting everything in balance. So again, it's not very surprising that this transcription factor, whose job is to regulate the expression of effector genes, has accommodates a large number of regulatory elements. And again, it was also very interesting to know that this particular region in human harbor a garden variety of genetic variants associated to autoimmune diseases. We also wondered whether this pattern of enrichment of genetic variants associated with diseases where immune cells are key players, most with a very specific focus of autoimmune diseases. Is this pattern true genome-wide, and can we really characterize this beyond BOC2? And this happens to be really the case. Again, I would like to highlight that the genetic variants associated with these diseases are events that happen germline, and this is the information that we extracted from population studies. And the super enhancer characterization is something that we are looking at the chromatin of these primary immune cells, and we really ask the question of, are these regions have any particular property or a feature compared to the genomic location of these genetic variants. And it was really interesting to see that this is really the case. So the super enhancer of T cells harbored a very large portion of, and a statistically significant portion of genetic variants associated to rheumatoid arthritis, IBD, type 1 diabetes, and, okay, this is getting worse, multiple sclerosis and type 1 diabetes, diseases that are known to, that T cells are key players. And again, genetic variants of these diseases were not enriched in the rest of enhancers that don't have this property. And in contrast, or as another kind of control, type 2 variants of type 2 diabetes and cancer, which again we specifically excluded any immune-related cancer for this particular analysis, were not enriched in the super enhancer regions of CD4 T cells. Again, highlighting that a very small change in these important regions, such as Manhattan, that a very small glitch in Manhattan can have a huge impact on the whole country, can really cause a lot of diseases and abnormalities. So what I told you so far was that by simply looking at the enhancer landscape in CD4 T cell subsets, we found tens of thousands of different regulatory elements, and also we look at a subset of them, super enhancers, and found interesting properties. But our other question was about who makes these changes happen. So is it about the category of transcription factors that are able to sense the changes in the cytokine environment, or is it about these more endogenous transcription factors that are expressed and then shape the enhancer landscapes afterward? We generated the map of P300 binding in the absence of each one of these transcription factors, and surprisingly, again, this was completely opposite of what we expected. The endogenous master regulators, the absence of endogenous master regulators, made some change in the profile of P300 binding, but not as much as the loss of stats. And this was done in Th1 cells. About 50% of the genomic regions bound by P300 lost P300 binding in stat deficient cells, STAT4 deficient cells, similar to in the case of Th1, it was about 75% of the Th2 specific enhancers were STAT6 dependent. Obviously, there was a reviewer's comment suggesting that, okay, this is so you lost stats, so you lost everything, the signaling wasn't really happening, and if you overexpress the master regulator in stat deficient cells, you might be able to recover all the enhancer elements. And this wasn't really the case. When we overexpressed T-bet in the STAT4 deficient cells, and we were able to fully recover interferon gamma expression, we were not able to recover P300 binding, again, suggesting that it's all about the signal transduction pathway, telling or recruiting all the proper enzymes to go after all these non-coding elements and add this further acetylation to these regions. So as I suggested, as I just highlighted, the changes in the signaling environment of cell is very linearly mirrored to the epigenome by signal transduction pathways and proteins like STATs. Within a span of a few months, this observation was seen. This was seen in other systems such as Th17 cells and Tregs and macrophages. So together with putting all these studies together, we would like to suggest that the formation of enhancer landscapes in immune cells seems to be a hierarchical event, and there seems to be different categories of transcription factors doing different kinds of jobs. The first group of transcription factors that can be called pioneer factors are able to have the property to bind to nucleosome. They don't need to bind to nucleosome-free regions. They bind to the nucleosomes, and they are able to open up the chromatin by recruiting the chromatin remodeling enzymes. And they further add these kind of poised marks, such as HTK4 monomethylation, and generate this landscape that will eventually be used by proteins like STATs that will add this further acetylation and activation mark. And the third category of transcription factors are proteins that are able to bind to a variety of regulatory elements, but maybe more specifically to promoters, as our study on T-bet and I think Rudensky's and FOXP3 really showed that the enrichment of these transcription factors are mostly on the promoters. And these proteins act as the final firing of expression at these genes. So together, again, as a summary, I showed you how, as kind of parallel to what Nick showed you, in CD4 T cells, also there seems to be a huge gain in the enhancer elements. And going back to our million enhancer possibility, it seems that every activation, every combination of cytokines really go after a distinct part of the genome for adding these kind of histone modifications or making the accessibility, making the chromatin to be accessible. But maybe the implication for the concept of immunotherapy is whether we can use these maps. If these maps are so digital and if we are able to gain such cell type specific, condition specific information from the enhancer landscape, whether we can use these maps within the context of tumor infiltrating lymphocytes and ask a kind of a reverse engineering question and say, what are the particular immune cells present in the samples, and are we able to learn more about the identity of cells that are present in a variety of tumors? And with that, I would like to thank my postdoctoral advisor, John O'Shea. This was obviously what I did in his lab. I was very fortunate to collaborate with a lot of investigators at the NIH. The work with super enhancer was in collaboration with Francis Collins' lab and Vittorio Sartorelli. My lab is very small, and we are recruiting computational postdocs. So if you are a computational person or you know anyone, please let me know. And I'm also collaborating with John Weary and other investigators at Penn, and I also like to thank the funders. And if you have, I think, any questions, we will be proceeding to the next session. Thank you.